Hello, everyone, and welcome to today's session on insect genomes, from chromosomes to ecosystems. I'm Kim Walden from the University of Illinois at Urbana-Champaign in the US, one of the session co-chairs. Rob Waterhouse from the University of Lausanne in Switzerland is also co-chairing this session. The format for today's session is a series of 5 pre-recorded presentations, followed by a brief Q&A session. Following the Q&A, there will be a spatial chat and formal discussion in the Insect Genomes room, and you can also join the BG21 Slack channel for further discussions. We ask that if you have questions for the presenters, please type your question in the Slido chat box below the video. Also, please include the speaker's name so that we can direct the questions appropriately. You can also upvote questions of interest. Lastly, please post your questions promptly so that we can collate them prior to the live Q. I'll now turn it over to Rob to introduce the speakers.
Thank you very much, Kim. So yes, we have from 4 different countries, 5 different speakers, and possibly some useful information about more than 6 different insect orders, starting with Kim, who will kick off with the EBP pilot project and the latest, then followed by Katie Brown from the University of Cambridge in the UK on identification of viral transcripts in RNA-seq datasets. Then we'll have Christina Gagalova from the University of British Columbia in Canada, talking about her work on the pine weevil, followed by Roddy Prakana from Queen Mary University in London, UK, on his fire ant work. And finally, with Miki Ballant from the Center for Translational Biodiversity Genomics in Germany, looking at shotgun metagenomics and taxonomy, biomass, and reference genome properties in soil invertebrate communities. So we have a cool program lined up and ready to take it away.
Hello, my name is Kim Walden. I'm an assembly analyst in the High Performance Biological Computing Group within the Roy J. Carver Beck Biotechnology Center at the University of Illinois at Urbana-Champaign in the US. I'd like to thank the conference organizers for giving me the opportunity to present our work from an Illinois pilot project as part of the larger Earth Biogenome Project. This pilot project was initiated by Gene Robinson, one of the founders of the Earth Biogenome Project, as well as by Jeff Stein and Chris Dietrich from the Natural History Survey and Prairie Research Institute. We were funded by the Illinois Innovation Network, a group of Illinois colleges and universities developing innovative projects to advance the economy and workforce in Illinois. The goals of the pilot project were to demonstrate the feasibility of rapidly sequencing and assembling 10 insect genomes to contribute to the Earth Biogenome Project, as well as the capability of our core sequencing facility and the bioinformatics team to produce 8 high-quality assemblies, while providing training to other scientists. The project kicked off in late summer 2020 with insect specimen collection, and genome assemblies were mostly completed by mid-spring 2021. We held a hybrid virtual in-person capstone workshop in July to present the assemblies and formulate strategies for larger scale funding. The insects selected for this project are native to central Illinois, which was historically a vast prairie ecosystem. But it is now a major agricultural area dominated mostly by a monoculture production of soy and maize. All of the insects selected have local agricultural relevance as either herbivores, predators, or parasitoids. The 10 samples selected represent the orders Lepidoptera, Coleoptera, Neuroptera, Diptera, Hymenoptera, and Hemiptera. Very little sequence information existed for these samples prior to the pilot project. And so one of the goals was to generate genomic information within a single ecosystem that could shed light on a variety of interactions. Chris Dietrich collected insects from the field, sorted them, identified them to species level when possible, and vouchered them at the Illinois Natural History Survey at the University of Illinois. Multiple specimens of each species were flash frozen at -80 for sequencing library preparation at the high throughput sequencing facility in our biotechnology center. DNA was isolated from only the insect head and thorax when possible, thereby avoiding the abdominal contents and the larger specimens. However, most of these species are less than 1 centimeter, so in some cases, the entire insect had to be homogenized. A modified C-tab extraction with an overnight proteinase K incubation protocol was used, followed by a QIAGEN MAGATRACT kit cleanup, which generated 6 nanograms to 5 micrograms of DNA per insect, with the majority of fragments longer than 40 kb. Current low input DNA library protocols have allowed for multiple types of sequencing libraries to be prepared from small specimens. And so the goal was to construct a total of 3 libraries. First, a linked short read TELSeq library to be used for genome size estimation using GenomeScope, and then downstream scaffolding, which is a novel use case in this project. The second library was the PacBio HiFi library, generating long, highly accurate reads. The GenomeScope analyses guided the number of HiFi flow cells needed for sequencing on our core facility's PacBio Sequel IIe. 2 of the samples, EB14 and EB20, were not pursued for PacBio HiFi sequencing at this point, as their estimated genome size would have required a number of flow cells outside of the pilot project budget. Unfortunately, in all cases, the DNA yield was low enough that a second insect was needed to prepare the Dovetail OmniC libraries for long-range scaffolding. The general assembly strategy for the pilot project was as follows. First, the PacBio HiFi reads were assembled with the HiFi assembler, which can produce partially phased genome assemblies. I should mention that our group tested multiple assemblers, determining that HiFi was the best assembler for our data. At all stages of the assembly process, the gene space completeness was assessed using BOOSCO, an orthologous gene prediction pipeline. After initial assembly, residual haplotigs were purged with the purge dupes tool. We then used the linked TELSeq reads again for a first round of scaffolding using the ARCS links package, and then further scaffolded to chromosome links using the Dovetail OmniC reads, the salsa scaffolder. Gaps were then filled where possible with the long read tool Cobbler and Rails and the short read tool Sealer, part of the Abyss assembler package. Downstream QC and curation, including detecting contaminant sequences, assessing misjoins, and finally annotating the genomes. The 8 assemblies from this pilot project all meet the Earth Biogenome Project guideline of contig N50 greater than 1 megabase and scaffold N50 greater than 5 megabases. In the case of EB29, a haploid male braconid parasitoid wasp, the main assembly is likely composed of entire chromosomes. We have detected several scaffold misjoins that artificially inflate the N50 values in 2 samples, and these will be corrected when additional funding is secured. The BOOSCO ortholog completeness scores for these 8 insect assemblies are excellent, with at least 90% of the Insecta ODB10 database orthologs detected. Less than 2% of the BOOSCO orthologs were not modeled for these genome assemblies. We evaluated the assemblies with other EBP recommended tools, such as Mercury, to assess genome completeness using K-mer analysis, and blob tools to identify contaminants. I could spend a whole talk discussing the blob tools, contaminant detection analyses, but the highlights are that we now have glimpses into potential prey items, pathogens, and endosymbionts for these insects. Several insect samples were initially identified to only the family level, and so we mined the assembled mitochondrial genomes and their encoded cytochrome oxidase 1 genes for species identification using the Barcode of Life database. We were able to assemble mitogenomes for all 10 insects using MitoFinder with the available references from NCBI and confirm their identity. One of the novel results was the identification of a prey species for sample EB5, the carabid beetle. The initial mitochondrial genome identified from the final purged assembly did not belong to the beetle, but instead to a chironomid fly, which was likely the last meal of the beetle before collection. We returned to the primary assembly prior to haplotig purging and correctly identified the beetle mitogenome. This is a great example of the advantages of isolating DNA from an entire insect and the disadvantages of isolating DNA from an entire insect. We annotated the assemblies using the Breaker2 pipeline with ab initio gene prediction using GeneMark and Augustus. In lieu of RNA-seq data, we opted to provide annotation hints with ProtHint using the arthropod orthologs from OrthoDB. The BOOSCO completeness scores for the predicted proteins are quite good for most of the samples, given the absence of RNA-seq data. We then added protein domain and functional annotation using InterProScan and SwissProt BLAST-P hits. We hope to make these annotation sets available soon, as they are providing rich data sets to mine. For example, genes involved with olfaction, gustation, and plant compound detoxification were predicted for EB1, the geometrid moth. In summary, this pilot project produced 8 high-quality insect genome assemblies, all of which exceeded EBP guidelines, using best practices for small wild-caught samples and employing novel uses of sequencing technologies. Ecosystem-level information was captured from viral, fungal, bacterial, and prey sequences. Future goals for this project are to secure additional funding for final curation and public deposition of these genomes, as well as to scale up the project to sequence many more genomes from an Illinois ecosystem. I'd like to acknowledge the pilot project PIs and the Illinois Innovation Network for funding. I'd also like to acknowledge the entire team that produced and managed the data, assembled the genomes, and supported our research cluster. And I'd like to thank you for your time listening to this presentation.
Hi everyone, my name's Katie Brown. I'm a postdoc in Andrew Firth's lab at the University of Cambridge in the virology division. And I'm just going to talk a bit about my project on identifying and characterizing RNA viruses in publicly available high throughput sequencing data. So my project's based on the premise that it's often possible to identify material derived from RNA viruses in publicly available RNA-seq data. And we found in our lab, and other groups have also shown, that this is actually the case regardless of the original research question that the data was generated to answer. So even with data sets that were RNA-seq data sets that were generated for questions which are nothing to do with virology, it's often possible to actually identify material and find out things about the RNA viruses in that species just by screening the RNA-seq data that's been generated for RNA viral reads. Now, there's over 2 million RNA-seq datasets now available for free publicly on the NCBI Sequence Read Archive, which is the database where all newly generated next-generation sequencing datasets should be deposited. So the aim of my project is essentially to develop a pipeline to screen these datasets, find material derived from RNA viruses, and then find out as much as I can about those viruses in that particular species. It's a fully computational pipeline, which has several steps. I want to download the data, identify reads which are potentially derived from a virus, combine these where possible into complete viral genomes, and then characterize the phylogenetic relationships between the species. And then the eventual plan with this outcome, if the output of this pipeline, is to look for novel viruses or viruses which are in unexpected hosts, to look at the relationships between viruses from different species, and to look at relationships between viruses from different species. So of course, it's not possible to immediately screen 2 million datasets. So we've then come up with a pilot project to help develop and then test my pipeline and hopefully find out something interesting. And this is on the identification and characterization of viruses, specifically in bees and associated arthropods. And that's what I'm going to talk about today. So why did we choose honeybees for this pilot project? So honeybees have been studied in much more detail than the majority of non-lab insect populations, and this is because of their massive importance to human food security and their economic importance as pollinators. And because they are so important, honeybee viruses are very well known, or reasonably well known. We know quite a lot about the RNA viruses of honeybees already. So this is a good starting point for my pipeline because we know something already about what we might find. However, honeybees don't live in isolation. They interact with many other species. This includes other species of pollinators like other bees and wasps, which they share flowers with. They also interact with ants. So because of the sugar in beehives, they're very often infested with ants. And bees are also known to be very often parasitized by mites. So the question for my pilot project is, are these honeybee viruses that we know about, are they limited to honeybees or are they more widespread? And the idea here is that although we consider them to be honeybee viruses, in at least some cases, this is likely just to be because these are the species that have been most widely screened. And it might actually be that these viruses are much more widespread. And we also want to know what other viruses are found in the non-honeybee species, because some of these species of other species of bees or of wasps and ants and mites have never been screened for viruses before. So there could well be other interesting viruses in there which haven't been discovered. So I want to take all of the publicly available datasets from any of these species as a subset, as an initial subset of RNA-seq datasets. And this adds up to almost 3,000 RNA-seq datasets. And this is all of the data which was available at the start of my project for the Hymenoptera, which is bees, wasps, ants, and sawflies, and then also the Acari, which is ticks and mites. I'm just going to run very quickly through my pipeline. So the input to my pipeline is unfiltered short RNA-seq reads from many species, and the output is either full length or partial viral genomes, and also some phylogenetic analysis, giving some idea of what the viruses in there might be. There's 6 main steps. I'm just going to talk briefly about each. So first, I pre-process the data, which involves downloading it and performing some quality control. And I end up after this step with 50 billion, from my 3,000 datasets, I end up with 50 billion high quality trimmed reads. So this is a massive amount of data that I then have to work with. So the next thing I want to do is filter the reads. And basically what I want to do here is remove anything that I know what it is and it isn't a virus, because I'm only at this stage interested in the RNA viral reads. So I mapped sequentially to these different databases and remove anything which maps well. And after this, I'm left with 15 billion what I call unknown reads, so reads that aren't from a known source. I then de novo assemble the reads using Trinity, and this is by far the most computationally and time-intensive step to actually then assemble all of these reads into contigs. But after this, I end up with about 100 million contigs across the different species of different lengths, which are not necessarily viruses, but which have the potential to be derived from viruses. I then need to filter them, so look for the ones which have some similarity to known viruses. And I do this using profile HMMs. So these are generated, we have these curated alignments for 77 different genera of RNA virus, and we base them on the RDRP gene. And we use this gene just because this is the only gene which is shared between all RNA viruses. So we use the RDRP gene alignments and this HMM model to then we compare this to all of the contigs and look for things which have significant similarity to RDRP. And after this, we have about 25,000 potential RDRP-like contigs remaining. I then want to identify any open reading frames in these contigs. I cluster them, and then I perform phylogenetic analysis, first assigning them to a major, so kind of rough analysis to get broadly what type of virus it is, and then a more specific analysis to assign them to a smaller cluster. And this leaves me with about 700 potential viral species from my initial 3,000 datasets. And this step's still ongoing, so I can't give exact numbers here, unfortunately, but I then perform genomic analysis where I try to, where possible, scaffold together the short contigs to try to get longer contigs, and also look for other viral genes using the same process of profile HMMs against the contigs. I'm just going to go briefly through a couple of results. So this is a phylogenetic tree for the IFLA viruses. So IFLA viruses is a major group of arthropod viruses, and it includes many known bee pathogens. So this is kind of a good starting point. On this is the known phylogeny prior to my study. So the black dots are all of these are arthropod viruses, and then the ones marked in blue are specifically in bees, wasps, ants, or ticks. So there's already quite a lot known in this group about viruses from these species that I'm interested in. However, if we then go to the results of my study, so I found over 100 clusters of IFLA viruses in my data. So if this is the known tree on the left, and then this much bigger tree on the right, where each pink dot is a virus that's been identified in one of these datasets in my study. So it's more than doubled what the number of known IFLA viruses in total. And these are all just from screening these public RNA-seq datasets. Then I'm just going to talk very briefly about one specific virus, deformed wing virus. So this is a major, well-known honeybee pathogen, and it is known in other arthropods, so it's not completely limited to honeybees. That has been seen in a few other species, but it's not considered to be widespread. However, if you look at my results here, so in this phylogeny, this line here is the number of datasets of different types where we found evidence of DWV, and we actually found it in 737 out of our 3,000 samples, including around 10% of ants, wasps, mites, and ticks, so massively more widespread than we expected, and in over 50% of the bee samples. And if we then look at the host, these are host phylogenetic trees where the red dots are the species where we found DWV, and it's really widespread across the phylogeny of all these different species. So just to conclude, RNA-seq datasets often contain viral RNA, and my pipeline can process them to identify viruses. We've found many novel and known viruses in these species. Cross-species transmissions appear to be common, but there's a huge amount of additional work left to do to fully characterize everything. And then I just want to acknowledge, especially my supervisor, Dr. Andrew Firth, and the rest of the Firth Lab, as well as our funding bodies and everyone that's provided us with compute. And thank you very much.
Okay, hello everyone. My name is Christina and I am a student at the UBC, which is in Canada, Vancouver. I'm part of the BTL lab, so we are working on mostly genome assembly methods, and we do also projects for genome assemblies, as the project that I'm going to talk about today. Today I'm going to talk about the spruce weevil, which is a very highly disruptive pest. And this species actually requires quite a lot of money for monitoring and especially containment for part of the forestry programs. As you can see from the picture on the left, the spruce weevil is quite widespread across the North American part. And originally it was thought that it was 3 different species, but now today we know that it's only 1 species that causes so much disruption. The spruce weevil can actually impact several conifers, and along those, the one that we mostly work with are the white Engelmann and Sitka spruce. There are, of course, other conifers that are impacted, but those are also in the States. And we have a growing catalog of species that are impacted. This is a picture of how the infestation of the spruce weevil looks like. The juvenile trees are basically not able to develop properly since the larvae is feeding on the apical shoots. Today, I'm going to talk about the genome assembly work that we did for this species. So we sequenced a single pupa from a wild collected sample here in Canada. So the reads are generated with 10x chromium link reads, and I used the standard pipeline for the assembly. So I used the Supernova, which is provided by 10x Genomics, and then carry on with the pipeline that has Porch Haplotics, and especially 3 software developed at my lab. So Tigment, Arcs, and Sealer. And I encourage you to take a look at the GitHub repo for more information. The assembly that we've got, it's still a draft assembly. So we have an NG50 of 87.7. And the good thing is that we are very close to the estimated size of the genome that we are sequencing. So the genome size, it's actually very big. It goes about 2 gigabytes per cent. We assemble 1.83. Together with that, we have a good Busco complete reconstruction. And on the top of that, we did a genome assembly, genome annotation, sorry. We also assembled, so my colleagues assembled the mitochondrial genome and did the annotation. And we found a significant amount of reads from the genomic sample of Wolbachia, which is an endosymbiont that grows, basically, it's part of the endosymbiont group of the spruce weevil. And we were able to assemble also the genome of the Wolbachia. All the resources that I'm listing here today are publicly available, and you can actually download them and use them for comparative genomics. As part of my project, I'm really interested in the biodiversity of this very large group of insects. They are part of the beetles, so coleopterans, and the curculionid is this very large family of 60,000 species. At the current day, there are like several genomes that are available. And only in 2020, 2021, there were 6 new genomes that has been released. So there is an increasing interest to study these species. The phylogeny here, which is created with a very large number of single copy genes, it's basically showing the relationship of all the available genomes that are released. You can see that there is a lot of variability, and like hosts are very different. And they both span new and old world, so Europe and the Americas. The spruce weevil on the top, it's part of the Molitini, only 1 species in this group. And I also want to point that we have also the genome of the Eurasian bark beetle, which is also of interest for the forestry in Europe. And this is actually the cousin of the mountain pine beetle, which is another species very important in America. So the spruce weevil has a lot of biodiversity, so a lot of species, but it has also very diverse genome features. So this is the phylogenetic tree that I just showed you. And this is the representation of the repeats as shown as a Kimura substitution level. And it's possible to see that there are like bursts and extinctions of repeats, especially for the species that are shown with the asterisk. Especially 4 species of those have a larger genome. So it's the case of the spruce weevil that has a 2 gigabase per genome. And this actually correlates with the burst of repeats that is quite recent. And it probably explains why the genome has become so large. So this is part of the biodiversity and the genetic features that we are studying. I just want to finish with a little bit of discussion of what is this going to be in the future. So we generated several resources that we can use for the analysis. We have a nuclear genome assembly, mitochondrial, and we started with the first Wolbachia and the symbiont genome assembly. So this is a start, this is a reference, and we would like to use that for especially analysis of population data. So several samples are going to be sampled for different populations of weevil that can infest different hosts and different conifers. And we are going to look at the biodiversity, the genomic diversity, their genome size, their genome characteristics. And especially we are going to look to the adaptation of the spruce weevil to the host and also the different geographic regions, because climate change is happening and this can of course impact the infestation of the spruce weevil, especially for conifers, which are really susceptible. And our programs consider this type of studies. And lastly, we would like to focus the studies on the Wolbachia and the symbiont. So as you may know, Wolbachia, it's a reproductive endosymbiont, and this can actually interfere with the reproductive cycle of the spruce weevil. And we would like to better understand their relationship. So not much is known about Wolbachia and the curculionid. And this can definitely be something to know more and like use as a tool to possibly control the infestation of the spruce weevil into the future. So with that, I would like to thank all my supervisors, all the people that give me mentorship to this project, and all the people that work on this project, especially Luca, Diana, Lauren, who did a great job with the assembly and the parts that we all work together. Thank you also to the CANSIC 150 and the Spruce Up. So please reach to me if you have any questions.
Hi, I'm Roddy. I'm a postdoc with Yannick Worm at Queen Mary University of London. Today I'm going to talk about dimorphism in social type in the Phaeran Solenopsis invicta. So this species of Phaeran has 2 different types of colony organization. So there's colonies with a single queen and colonies with multiple queens, and they differ in a number of physiological and behavioral traits. It's important to say that single queen colony organization is the ancestral social form to all ants, whereas the multiple queen colony has evolved independently in different species, different points of the ant phylogeny. In the fire ant, this dimorphism is controlled by a supergene. So supergenes are chromosomal regions with some sort of repressed recombination that allows different variants of the chromosome to coexist in the population. So each variant will have a different combination of alleles, and each allele will contribute to a different morph in the population. In the case of the fire ant, single queen colonies have the big B variant of the supergene, so all individuals have big B in the colony, whereas the derived form, the multiple queen colony, has a derived form of the supergene, so the little b version. And these colonies, all queens have the little b allele. They carry at least one copy, and most workers too. And what happens is these colonies accept incoming queens unless they don't carry this little b allele, in which case they will kill any incoming queen. Right, so this is a very large supergene. It extends up to about 27.5 megabases. So that's over half of chromosome 16 of the fire ant genome. And this estimate includes non-assembled parts of the chromosome. This supergene has about 470 protein coding genes, although we don't know which, if any, of these genes actually affect the phenotypes that are under control of this supergene region. The last thing is that this supergene occurs in multiple species of fire ants in the Solenopsis genus. So both Invicta and Rishtri here have multiple queen colonies, and in both species, the multiple queen colonies are associated with this little b variant of the supergene. So in summary, I've told you that fire ants have a social supergene that controls a complex social dimorphism in colony organization. The supergene spans a very large genomic region, and it occurs in multiple species of Solenopsis ants. And the question in this talk is where in the phylogeny is the origin of this little b variant, the derived variant of the supergene? We start with a very simple hypothesis, which is that there is a single ancestral origin of the supergene, which occurred before the speciation of the species that are socially dimorphic. And we test this by looking at genomes. So we've looked at 368 whole genome sequences from 8 species of fire ants. These samples have been taken from all over South America, either by us or from previous studies by us and other authors. Most of the samples are haploid because male ants are haploid. We have used freebase to call SNPs between these individuals, and we only looked at single copy genes to avoid erroneous calls in collapsed bits of the genome that are repetitive. So the fire ant genome is very repetitive, and we want to avoid collapse repeats causing false SNPs. So we only looked at around 1,700 single copy genes. For each gene, we made a phylogenetic tree, and then we made 2 consensus trees from these genes. So one tree used the 97 genes that map to the supergene. So these are the supergene tree. And the other tree uses all the other genes that map outside the supergene. So this represents the species tree. Okay, the results are shown in this plot. This is quite complicated, so I'll go little by little. So if we first look at the species tree on the left, here on the right is a simplified version of the figure. We see that all S. invicta samples form a single monophyletic clade, and all S. rishtri samples form another single monophyletic clade, and then each outgroup species also forms a single monophyletic clade. So far so good. But when we look at the supergene tree, what we see is that all the little b individuals, regardless of species, they all clustered together, separated from all the other samples. This means that there is a single origin of little b in the Solenopsis genus. If we look more closely, we see that actually the little b, all the little b samples form a sister clade to the big B samples of S. invicta. And what this means is that little b seems to have evolved within S. invicta, and that means it's been after the speciation between Invicta and Rishtri. If it was before the speciation, the separation between big B and little b would be before, here on the tree. So to put it in another way, we don't have a single ancestral origin of little b, which was our hypothesis. Instead, we have little b emerging within S. invicta and then introgressing into S. rishtri. Okay, it's a bit more complicated than this because within these little b samples, there's some dark blue samples. And if we compare the supergene tree with the species tree, these dark blue samples have diagonal lines all the way down here. So these are samples that belong to the outgroup species but which carry the little b variant. And what this tells us is that the supergene not only introgressed from S. invicta to S. richteri, but it also introgressed from S. invicta into 3 other Solenopsis species that we didn't know were socially dimorphic. So how do you get these supergenes jumping between species? So we think it's because of introgressive hybridization. So hybridization occurs when individuals from different species hybridize and allow jumping of alleles between species. And it's really interesting when these alleles are adaptive, because it means that a phenotype can evolve in one species and jump to another species, as is the case here for coloration in a heliconius butterfly. So what are the implications of our work? So we show that we show a striking example of introgression of a supergene that is very large and which is associated with a complex behavior phenotype. And if you think of the introgression of a very large supergene, then raises the question whether this supergene is in any way incompatible with the host genome, and whether there has been any evolution to resolve those incompatibilities. The other implication is that the polymorphism has remained balanced in the host species after the introgression. Right, our work is published as a preprint on bioRxiv. You can take a look at that, and thank you for listening.
Hi everyone, I'm Mickey Balint from the Löwe Center for Translational Biodiversity Genomics. And I'm very happy to tell you more about our experiences with the metagenomics of soil invertebrate communities. So, soil biodiversity is important, but what can be in soils? What you see here is a spoon of soil, and what you might find in there is several billions of bacterial cells. We can find hundreds of meters of fungal hyphae. But what I'm going to talk about today is actually the invertebrates, the tiny animals. There can be many, many different taxa in only a handful of soil. So today I'm focusing on rather on the smaller ones of these taxa, especially on springtails, mites, nematodes, and potworms. So if somebody wants to answer questions about soil biodiversity, it is important to understand the composition and the function of these communities. And here, regarding especially the function, the biomass of the organisms is very important, because species with a higher biomass have a good chance that they have a stronger impact on the functioning of the soil than species with a lower biomass. So we need methods which are able to identify species, but they are also able to identify the biomass of these species. The obvious choice going for community composition and molecular identification would be metabarcoding. So this is a conceptually fairly simple procedure. One is taking a sample, is extracting DNA from it, does a PCR on a taxonomic marker gene, does then some high throughput sequencing on the amplicons, and after the bioinformatics gets species by sample matrix. Now the problem with this is, or might be, the PCR, which can be highly taxon specific. So the efficiency of the PCR might depend on the taxonomic identity of the organisms, and some species might have many more reads in the resulting data set than others, because the PCR primers are some kind of biased. The other option is shotgun metagenomics, which is a very simple procedure, except that we are missing the PCR step. So theoretically, shotgun metagenomics should be able to deliver us a better biomass read relationship. So this is what we set up ourselves for. We try to understand how shotgun metagenomics records species from a mock community, how it records biomass read relationships, and to investigate what might influence biomass read relationships. So at the front, I want to also say that soil invertebrates are kind of excellent targets for metagenomics. What you see on the left is extraction device for soil invertebrates. And now I want to specify that this is an extraction device of the animal specimens, not the DNA. How it functions, people put soil into these funnels, then they apply heat from the top, and then the soil invertebrates, especially the microarthropods, have a reaction to this. Once the soil is trying to dry out in the top, they start crawling down into more humid lower regions. But this apparatus at the very end has a vial with some preserving liquid in, so the animals kind of nicely crawl into this liquid. And this is something we can immediately use for metagenomics. And we can scale this up, and like with this McFadden apparatus on the right, you can actually extract hundreds of soil samples at once. And once you do this specimen extraction, what you get is a sample like this. This is a Petri dish where all those dots are animals that were crawling out from one of these soil cores. Now we can take this sample and simply crush it relatively easily. We don't need big devices for it, and do DNA extraction on it. So the mock communities we worked with, these are the result of a tremendous work from my colleagues from the Senckenberg Soil Zoology Division from Görlitz. So they're also co-authors on this presentation. They were taking 28 species and created 4 different mock types with different biomass relationships. And replicated them 3 times. So that's a tremendous work. Some of these species have to have a huge number of specimens to give these biomass values. Some species, specimens, we actually have to cut them to kind of create these mocks. So many thanks again for this work. What we're doing in terms of methods, DNA extraction with a pistil Eppendorf crushing, QIAGEN blood and tissue, library preparation with the NAPDEX Ultra, sequencing on an Illumina platform. The bioinformatics consisted of read trimming and metagenomic assignments. And in this context, it is important that we generated an own database of over 270 soil invertebrate species. We have quite a number of organism groups typical for soils, but most of the genomes are coming from springtails, myriapods, and mites. Most of these genomes are done from single individuals. And what we are getting at first with terms of species detection, what you see on this graph is on the y-axis is the Kraken classification threshold, and on the, this is the x-axis, and on the y-axis you see the percentage of species which get correctly identified in green, or they are false negative identifications or false positive identifications. We have a number of consistently false negative species. Most of the species are recovered regardless of the threshold, but there is a tremendous drop in the false positive identifications. Nonetheless, even with very stringent classification thresholds, we have a number of species that are false positive identified. And we think this is because of the genomes which were not repeat masked in this phase. The second result is the biomass read relationship. Again, on the x-axis, you see the biomass, and the y-axis is a number of assigned reads on a log scale. The first thing what is really striking here is that we see a strong difference among the different taxa with respect to assigned reads. So the springtails are working excellently. They attract a lot of reads. The potworms are working quite poorly. We think that this big difference is coming from the DNA extraction. So probably some of these species release more or less DNA depending on their taxonomic identity. And because of the crushing that we did. We think that we should improve this. So the next thing that we are going to experiment here is improving crushing probably with steel beads or freezing. Nonetheless, despite of this very strong taxonomic effect, we see that there is a consistent biomass read relationship for most of the species. With higher biomasses, we are getting more assigned reads. Then we looked into the effects of genomes from the assignment database on the taxonomic assignment. What we find here is that genome completeness, in our case, had no strong effect for most of the organism groups on the number of assigned reads. With genome completeness, here we mean BUSCO completeness scores. There is, so the reason for this is like that most of the genomes that we are using here are fairly complete. They have high Busco scores, except one species of an Enchytraeus, which is decaploid. And in this case, the genome is not really complete. And we see that the genome completeness is actually strongly positive. Then we looked into the influence of the genome size. The expectation here is that organisms with large genome sizes will attract more reads from the metagenomic bulk extracts, and this is exactly what we are getting. So genome size is something that needs to be considered in metagenomic read assignments. And finally, if we are ordering the importance of these variables on the number of assigned reads, what we find is that the strongest effect, at least in this experiment, was the taxonomic identity. This is followed by genome size and then by biomass, and finally by the genome completeness down here, which is not strongly significant. I mean, statistically significant. The other relationships are statistically significant. Nonetheless, despite this huge taxonomic effect, we find that the metagenomic reads still represent the differences among the mock community replicates. So they are kind of still able to record ecological information on community composition, despite all the strong effects from the other factors. So to quickly sum it up, we see a strong effect on, we see a lot of false positives, which need to be addressed and checked where they are coming from. We see a consistent biomass-read relationship, but there are a number of factors which need to be considered, such as taxon identity and genome completeness and the genome size. And we see that the metagenomic reads can very nicely represent community composition in an ecological analysis. And with this, I want to thank you for our partners and funders, and I'm looking for questions. Thank you.
Hey, well, thank you very much, everyone, for the excellent talks and the questions coming in through the Slido. Perhaps we could just cycle through and try to get one question first for each of the speakers, and then see what we've got left to make up the rest of the time. So to kick off with the first speaker, with my co-chair, Kim, perhaps as more and more of us are embarking on these kinds of pilot projects like you described, for example, like the European Reference Genome Atlas pilot project, have you, in your team, have you collected some lessons learned, and do you intend to share them, and how?
Thanks. We do intend to share the output of these assemblies, and we are in the stages of finalizing some grant proposals to secure additional funding so that we can manually curate those assemblies and then deposit them publicly into NCBI. We also are working on a GitHub page where we can make available our pipelines and procedures that we used. From lessons learned, I think that ideally we would like to have RNA-seq data. And in this project, this pilot project, we did not generate that. So I think with small samples, that is still a challenge. But in the future, we would hope to be able to do that, to isolate RNA from the samples to support the annotation of these samples.
Okay, so that's an important lesson learned there. When you've got everything in place to get a beautiful genome assembly, but not yet the data required to then get the perfect annotations or decent annotations afterwards. That's
correct. We used arthropod hints from OrthoDB in the pipeline for annotation. So the annotations looked quite good despite the lack of RNA-seq data, but that's definitely an area that could be improved in the future.
Okay, thanks. I'll leave you to ask the next question of Katie.
Okay, Katie, this is from Rob Waterhouse. With the vast amount of data available in NCBI SRA, how do you go about practically selecting which SRA projects that you're going to use? Do you have a set of tools or criteria since there's such an abundance of information? I think you're muted.
Oh, I muted and then unmuted. Sorry. So we don't particularly filter data sets, we search. We basically take everything that matches. So we did pick the species of interest in different kind of smaller areas, and then we just to start with take every data set from that species and then only filter out if they're particularly poor. So we only filter out if the reads aren't recognizable, or if they're all ends, or if they have less than 100,000 reads. So almost only if they're really, really poor quality. And then from there, we assemble them, look for viruses, and then only at the end, once we found the viruses, we then go back and check that there was nothing. We then, where we find something interesting, we then go back and look at the input and see exactly where it's come from. And it's just easier to filter in that direction. That we find viruses, lots of the data sets, we won't find anything. So rather than doing the filtering at the beginning, we kind of do the filtering at the end once we know what's in there. So we don't really filter is the short answer.
Okay, great. Thank you. I'll let Rob ask the next question.
Yep. So now for Christina, we were, I think, quite a few of us interested in this repeat content and the variation that you found. And so the main questions coming up are based on, like, how did you go about characterizing the repeats, given that you're probably finding some novel ones, and what sort of proportion were completely novel, completely unknown? So methodologically and also like biologically.
Yeah, I suggest to run a more extensive pipeline that uses homology base, for example, or uses other databases. So I ran the most comprehensive one that I found, the EDTA, and there is a component of de novo discovery of repeats. So the most interesting thing I found for these genomes, especially for the largest one, so the spruce weevil and the Easter egg weevil, is that there is plenty of new repeats. So the genome, I think it's about 10 to 15% of the genome is covered by unknown repeats, which is remarkable, like for the biggest one. So we still have quite a lot of repeats to learn and to characterize.
And just to follow on it, those novel repeats, are they, I mean, they are lines or they're signs, or are they low complexity repeats?
I think it's totally uncharacterized. So they are not sign and line for sure, and not simple repeats. So I haven't dig into deep, like still work to be done, but definitely new classes of repeats.
Okay, thank you. It's really interesting to see that variation. Yeah. Go ahead, Kim.
All right, the next question is for Roddy from Rob Waterhouse. How did you technically achieve the consensus trees? Was it a concatenation approach or a multi-tree maximum support approach or something else entirely?
Right, so I think that the difficulty we had is that these two species are divergent quite recently, so maybe about 1 million years ago. But that gives us quite a shallow tree. And when you have that, things like incomplete linear sorting becomes a problem. So if we compare lots of different genes, each gene gives us basically a different gene tree. So to compare them, we use this method called Astral, which basically does very clever stuff that I can't really explain easily, but tries and gives you a consensus while controlling for the discordance between all the different genes. Yeah.
Thank you. Perhaps one more question?
Yep. Okay. So for Miki, we have a question here from Chris Laumer, who appreciates your talk as well. I found it a bit strange that the potworms had the poorest biomass read relationship. Shouldn't these soft-bodied organisms be even easier to lyse than the arthropods? A bit surprised to hear you think they need bead beating. Could it just be that they are more watery? I guess this is what's more squishable, yes?
Yeah, right. Thanks, Chris, for the question. I guess you're right with the potworms. In that case, it's probably not the problem that we cannot destroy them well enough. Nonetheless, we see a lot of variation still in hard-bodied things like the springtails and the mites. And for that, we are considering bead beating now. Looks like the pistil destruction approach was not enough. At least that's my impression.
Okay, thank you very much. So given that we're like 4 minutes over time, I don't know, should we be naughty and go for another round of questions, Kim, or should we be well behaved and save them for the spatial chat afterwards?
Yeah, why don't we do defer questions to the spatial chat session and to the Slack channel?
Okay, yeah, I'm good with that. So thank you very much to all of our speakers and all of you for joining us this evening. And a reminder that to join the spatial chat is really simple. From the platform, you can click through from network to spatial chat, sign in, or not sign in, just add your name, and then you can join the platform. And then I think Insect Genomics is somewhere down at the bottom of the list. So just scroll down a bit to find us, and we should be there shortly. So thank you very much, everyone. And thank you, Kim, for co-chairing with me, and maybe see some of you soon.